primarily in the probiotic and direct-fed microbial area. Dr. Billy M. Hargis is a professor at the University of Arkansas and has been working in this field for the past 25 years. Dr. Hargis is a professor. He's also the director of the Poultry Health Lab at the University of Arkansas, and he's also the Tyson Endowed Chair for Sustainable Poultry Health in the United States, a very prestigious position. So it's my honor and my privilege to introduce Dr. Billy Hargis for this morning's presentation. And again, thank you for coming.
So one of the questions, can you hear me? Everyone can hear me without the microphone? Yes? No? Yeah. Okay. One of the questions is, why are we worried about these things? Well, we're seeing antibiotic growth promoters disappear worldwide now. They're declining very, very quickly, and they're very important historically for meat.
Bill, you need your microphone.
Well, I asked the question. So, historically, antibiotic growth promoters have been very, very important for improving feed efficiency. With rising grain prices and rising cost of producing meat and poultry, we're going to have to find some alternatives to these traditional antibiotics as they continue to disappear. I bring you greetings from my institution, the University of Arkansas. This is the Center of Excellence for Poultry Science up at the top. And this is our laboratory down here at the bottom. It's located about 5 kilometers from the campus. They don't let people that do what we do work on the campus. We have a large group in our laboratory. There's currently 5 PhD level scientists in our group. We've had a number of PhD students just in the last few years that have contributed to the work that I'm going to be talking about, and 6 current graduate students that are working on this. There's approximately 20 scientists, postdocs, graduate students, and technicians in our group, and our goal is what's called sustainable poultry health. And what we're talking about is, what are we going to do when the traditional chemicals and drugs are no longer available for use? We're not arguing that they should disappear, but we certainly are predicting and anticipating that these chemicals and antibiotics are disappearing. Our mission is to develop novel vaccine strategies and strategies to combat the emergence of escape mutants with vaccination. One of the problems that we're having with vaccines globally is these serotypes continue to split. So a large part of our work is involved with vaccine production and new approaches for recombinant and autogenous vaccines. And a major effort in our group has been the development of highly effective probiotic and direct-fed microbials for the global poultry industry. We have contracts, grants, and collaborates with a large number of integrators and allied industry each year. So we work very closely with industry in our group. The ecosystem of the intestinal tract is phenomenal. These are big numbers, 10 to the 12th to 10 to the 13th. This is a 1 with 12 to 13 zeros behind it. That's how many bacteria are in 1 gram of the ingesta within the gastrointestinal tract of a chicken or a human. It's about 10 times more cells in the gastrointestinal tract, bacteria mostly, than the animal has for somatic cells. So the genome is huge, and these organisms have co-evolved with vertebrate animals over hundreds of millions of years. And there's a homeostasis here that's very critical. We're a tube within a tube, and this competition in the gastrointestinal tract. We're separated as humans, vertebrate animals, chickens. We're separated from feces by a single cell layer of intestinal mucosa, okay? What's going on in the lumen, the intestinal content, in terms of beneficial microflora versus harmful microflora is incredibly important. The beneficial bacteria directly compete with pathogens, and that's been very well established. This homeostasis that develops is incredibly important. If we take gnotobiotic animals, and we put these animals in a sterile environment and do not allow them to become colonized with intestinal microflora, the villi do not develop, absorptive capacity does not develop, the acquired and innate immune function of the intestinal tract does not develop, and these animals become exquisitely sensitive to even the mildest of opportunistic pathogens, and they'll succumb to lethal infections very, very easily. The intestinal tract is the largest acquired immune system organ in the body. There's more immune cells in the intestinal tract of a chicken than there is in the spleen, the bursa, the liver, and the thymus combined. Okay, so it's a very important organ for health, and these things do not develop without the appropriate colonization with bacteria. These metabolites that from fermentation of bacteria within the gut directly stimulate not only development but also uptake of nutrients, detoxification of toxic compounds that are in the ingesta, a remediation of ammonia that's generated during proteolysis within the gut. Very, very important for the health of the animal. This is an electron photomicrograph that we took a few years ago. This is a chick at 24 hours that's been placed in a clean environment. This is from the SICA, and this is a scanning electron photomicrograph. And you can see very few bacteria colonizing, and you also see that there's no development of the folds of the cecum for absorptive capacity. And this is a chick that's been treated with appropriate competitive exclusion type product, where you have actual colonization and you can actually see the folds starting to develop in 24 hours when appropriate microflora are applied. This is a very, very important concept, and it's something that I think we often forget. If we look at the infected dose 50, how many pathogens it takes to infect an animal, you can think of this as resistance to infection. For many enteric pathogens, opportunistic pathogens, during the first 10 days of life or so, you have an increase in the resistance to infection, mostly due to microflora and how the microflora interface with the host, and it's huge. The number, it takes about 10,000 times more pathogens to infect a bird at 10 days than it does at hatch. Okay, so it's an incredibly important process. And one of the concept has been, and we've got, these are cartoons, but there's data that actually support this, we're pushing this backwards and accelerating the onset. But it's not just that. When you look at what happens in the life of a chicken, okay, a broiler or a leghorn, there are many things that disturb the gastrointestinal microflora of the animal during the life of the bird. We have changes in diet substrates, least cost feed formulation, where we change from one substrate to another. We have no regard for what's happening to the microbial ecology within the gut. We see transient diarrhea, flushing, wet litter, when we have major substrate changes in the diet, for example, or if we apply a therapeutic antibiotic, which is necessary sometimes. We destroy all of the sensitive microflora within the gastrointestinal tract. Unless they're resistant, they disappear when we apply a therapeutic dose of fluoroquinolone, for example, to treat a mycoplasma problem. We destroy everything within the gastrointestinal tract that is sensitive to that drug. Okay? Who survives? Those organisms that are resistant. Which organisms are most likely to be resistant? Pathogens that have seen that antibiotic before. So what happens is we see this resistance plummet when we put these animals on a course. We published two papers on this a few years ago showing that the resistance plummets when we apply a broad-spectrum antibiotic to birds that have developed a very high degree of gastrointestinal resistance. And the opportunity then is, instead of allowing this to come back gradually over time, is to accelerate this, to push this back this direction. And I'm going to show you some data that indicates that that's possible. We have been focusing over the last 12 years in our laboratory at the University of Arkansas, and for more than a decade before that at Texas A&M, on developing effective, very effective probiotic, direct-fed microbial type of products. And it's not been an easy task. We have literally screened tens of millions of organisms over the years to not find a single organism that comes from a yogurt lactobacillus, for example, that's adapted to growing in milk at high temperature, but to identify organisms that are adapted to the gastrointestinal tract of the chicken, which can effectively compete with pathogens and can effectively stimulate responses in the animal, defensive responses. We're looking for effective colonization and persistence within the gastrointestinal tract. We look for direct exclusion of pathogens such as E. coli, Salmonella, Clostridium, and others. And new technologies are focused on the reduction of inappropriate inflammation. This is something I'm not going to be able to talk about right now. I don't have enough time. But we think that inflammation, reduced inappropriate inflammation, is one of the common themes of the subtherapeutic antibiotics in terms of how they improve feed efficiency, because in many cases they're applied at some sub-MIC levels. They're below levels that are required to kill the pathogens that are within the gut, and yet they still improve performance. And we're also working on some new candidates that I'll touch on a little bit later. Now, direct-fed microbials and probiotics have a terrible reputation. One of the biggest problems, in my opinion, is there's very little regulation of these products. So anyone can put anything in a bag or a bottle or a sack and say it's a direct-fed microbial or a probiotic. And it's caused a lot of issues with disregarding the possibility of being able to do really good things. So I wish there was more regulation of these. We've looked at many products that are actually sterile, that contain no bacteria whatsoever. They don't contain the bacteria that they say they have. They don't have bacteria that will even grow at the body temperature of a chicken. And it's no wonder that many producers are skeptical of probiotic type products. Antimicrobials and disinfectants interfere with direct-fed microbials or probiotics when applied in the drinking water. Residues of antibiotics or high levels of chlorine in the drinking water during application, not once colonization occurs, but during the application process, they can compete. Some effective organisms are not compatible. It's a big problem. As we screened and threw away millions of organisms and refined this to smaller and smaller groups of organisms, we found that we had candidates that were individually effective, but in combination, they were actually not effective. Okay, they were antagonistic to each other. And so you think that you've got a good one here, A, and you have B, that's a good one. You put them together and they should be at least the same, if not additive or synergistic. And we found that in many cases that was not the case. We had to look for those that were compatible as well. A relatively high dosage, much like that infected dose 50 curve that I showed you before. You have to have enough organisms that hit the gut all at one time to be able to establish the culture. And that's really critical. And diluting effective probiotics past the point where they're effective, doing research at one level of inclusion, and then marketing the same culture, but at 10 times to 100 times less concentration, it doesn't make any sense. I mean, can you imagine using an antibiotic, a growth-promoting antibiotic, at 1/100th of the labeled dose? I mean, they don't work. There's also sometimes a need for readministration after a gastrointestinal disturbance. And so it's both a tool, but these are the failures that we've seen historically with probiotic development, that we focused on how do we avoid these failures as we try to develop the next generation of direct-fed microbials. The potential is huge. And I'm going to show you data that says we can increase feed efficiency, we can reduce dysbacteriosis, necrotic enteritis, colibacillosis, salmonellosis, and so forth. We can reduce wet droppings within a 24-hour period, and we end up with drier litter very, very quickly. So what are the characteristics that are desirable for a competitive exclusion direct-fed microbial probiotic type of production? It must be fairly inexpensive to produce. We have to have batch fermentation. We believe that they should be therapeutically efficacious. You should be able to displace pathogens once they're colonized, rather than simply prophylactic efficacy where we try to protect from challenge. That's relatively easy. If you're looking for the rock star candidates, you're looking for organisms that can actually displace pathogens that have already colonized the gastrointestinal tract. We need things that are highly effective under frequent feeding and management systems, okay? And they must be defined and generally recognized as safe. And that has been the rule, at least in all developed countries. We had a breakthrough in our program in 2003 where we developed an in vitro system that was predictive, not perfectly correlated, but we could eliminate 99.9% of candidates that would not be effective when we took it to animal challenge. And we've refined the system since 2003. We've used this extensively in development of these lactic acid and bacillus-based products that I'm going to tell you about. We eventually, looking at the lactic acid bacteria are of the genus or related to lactobacillus. They may include Pediococcus, Weissella, lactobacillus, and there's several others that fall within that group. Many of these are generally regarded by safe by major regulatory agencies, and there's very little evidence that any of the lactic acid bacteria are pathogens, with maybe one exception. A specific combination that was developed in our laboratory was named B11. It was in the combination in our lab books was B11, and now this product is licensed by the University of Arkansas and produced by Pacific Vet Group, and it's called Floramax B11. And this is what I'm going to be talking about here at first. There's a lot of publications. I don't have time to talk about all these. These are available in PDF format on the Pacific Vet Group website, but there's at least 26 just for Floramax alone. And you can read about the scientific work that we've done in our lab. There's more than 100 published abstracts and presentations, and currently used commercially in more than 15 countries for salmonella control, enteritis control, production improvements, and prevention of necrotic enteritis. Okay, so as we've refined this, we found that the lactic acid bacterial candidates that we have included in this first lactic acid bacterial population actually produce bacteriocins and actually compete directly with pathogens, producing zones of inhibition much like an antibiotic. These are due to lactobacillus or bacteriocins. They produce hydrogen peroxide. They produce biosurfactants and organic acids, all which compete with opportunistic pathogens such as salmonella and clostridium within the gastrointestinal tract. And more recently, with microarray analysis, we've actually shown that Floramax actually decreases the innate immune response when animals are challenged with a pathogen. So we're thinking this may be a common theme with the antibiotic growth promoters. It's kind of exciting work. Some of the earlier work was involved with a Salmonella enteritidis. This is a very highly invasive Salmonella enteritidis that we use in our laboratory. And within 24 hours, we saw major reductions in colonization with Salmonella. We've looked at many, many serotypes over the years. It's not serotype specific. It works with E. coli, it works with Clostridium perfringens, and any other Salmonella. This is Salmonella Heidelberg, and we saw 80% colonization down to practically zero within 24 hours in multiple studies. One of the papers that was published in Poultry Science a few years ago is really startling, and we use Salmonella as a model. It's not because we care just about food safety, although we do care about food safety, but the Salmonella is a very good model enteric pathogen because it does reduce feed efficiency. When we challenge animals with a Salmonella in a clean environment, we can see several point reductions in feed efficiency just from the Salmonella challenge. And E. coli is very closely related to Salmonella, the avian pathogenic E. coli. But what's interesting about this paper that was published is that when we challenged animals and then treated with Floramax, within 12 hours of administration of Floramax, we saw major reductions in Salmonella colonies. These are logs, so this is 2.5 logs down to about 1.5 logs. That's a 10-fold reduction in the treated birds just within 12 hours. And then it continues and continues as the untreated controls continue to go up. So it's a very rapid response. Interestingly, we see anti-inflammatory activity within the gut approximately the same time point. We've also looked at things like necrotic enteritis in a laboratory challenge model that we developed. It's been a very reproducible challenge model. And this a very severe challenge. Mortality is represented here by these percentages, and we killed 47% of the control birds with this challenge. So this is a very severe necrotic enteritis challenge model. And we compared to BMD, bacitracin, at 50 grams per ton, which is a great drug for controlling necrotic enteritis. And that did significantly improve performance and reduce the mortality in that group. But the Floramax actually did better than the antibiotic. We have actually compared these cultures to very well-established antibiotic growth promoters in a number of studies and won. Why did it reduce necrotic enteritis? Using real-time RT-PCR, we were able to measure Clostridium alpha-toxigenic Clostridium perfringens specifically, and we were up around 10 logs of Clostridium perfringens in the untreated control, where we had 47% mortality. Okay, there's no wonder. 10 logs is a lot of bacteria. Bacitracin reduced by 100-fold. That's a 100-fold reduction. Okay, so bacitracin is a good drug. But here, we're talking about from 10 to the 10th to 10 to the 6th, that's a 4-log reduction, right? That's a 10,000-fold reduction in numbers of Clostridium perfringens within the gut. We ran a study that was published in Journal of Applied Poultry Research back in 2005 with an idiopathic enteritis problem that was occurring in turkey poults. This is very interesting because we knew that it was a viral etiology. The primary cause is a virus, but the company that we were working with, the integrator that we were working with, was finding that therapeutic antibiotic administration was helping. Well, that's pretty common because secondary bacterial infections are very common with viral enteritis problems. And so they were improving performance when they would treat this with antibiotics, even though they pretty much knew, we all knew, that the problem was not bacterial in origin, but viral in origin with secondary opportunistic pathogen involvement. And so using pens within the turkey house, multiple pens, replicates of 4 for each treatment, we were able to take untreated animals. We used the antibiotic treatment regime that the company was using, and we compared that to Floramax B11 and looked at body weight at day 21. And we actually beat a very aggressive antibiotic treatment scheme in this manuscript. This is a commercial turkey field trial that consisted of a very large number of lots of turkeys. And this is a very interesting slide for me because this shows you a couple things. When you randomly assign treatments within service technician areas across a complex, so they're absolutely randomly selected application of treatment versus controls. And we were able to do this because this product is delivered in the drinking water. You get enough data that you can start talking about it. Many people will do paired house trials to try to use 2 houses on the same farm. Well, there are huge factors within the environment of 2 different houses, even on the same feed, that impact performance tremendously. In many cases, the actual chick source for the 2 houses came from different parent flocks. So it's not until you start looking at the average of the treated versus the non-treated. We have a few untreated that did pretty well, and we have a few treated that didn't do so well. But you start to see this, and when you collect the data, you're talking about very significant increases in body weight, very big differences in average daily gain. Feed conversion was numerically reduced, and the actual cost of production was significantly reduced in that study, which was published. We followed this up with a commercial broiler field trial. There were 12 treated and 12 control operations on this project, and comparing directly the body weight of treated versus untreated, and average daily gain was staggering. So really important. The feed conversion rate was significantly improved, as was mortality reduction in the Floramax treated group in that published study. The economic return that the integrator reported was a 15-to-1 return on investment with that product. And we're very, very proud of this product that was developed in our laboratory because it is very effective. One of the advantages is that it can be applied at the discretion of the live production manager or the veterinarian in the field whenever it's needed. It doesn't have to go in the feed. But there are limitations. One of the limitations is that it has to be applied correctly. You have to train the producers to use it correctly and so forth. You don't have full control by the integrator. So some of the issues and limitations of these lactic acid bacterial products is shelf life and stability. If they get too hot, they can't go in the feed, and if they're applied correctly. So there are many problems, but there's huge opportunities. And you know, many of these problems, you say, oh, that's really troublesome. That's not true with Tylosin. Well, actually, they're much more stable than most of the vaccines that we use live in the poultry industry. You just have to have some education as to how they should be used. But still, we wanted to develop a spore-based probiotic, and we worked for a number of years on identifying effective bacillus candidates that are spore formers that would provide similar, if not better, activity when incorporated into the feed. We were looking for isolates that were heat stable that could go through the pelleting process. Okay. These are vegetative bacilli here, and when they're stressed, they form these endospores that are very refractive in this photomicrograph. And some strains, not all, are very heat resistant and very, very resistant to time. The shelf life is many, many years for these guys. Very tough in some cases, not all, but some. So our initial criteria for isolate selection with the bacillus product called sporulin was it must produce heat-stable spores. We needed a minimal loss due to pelleting conditions, must grow to high numbers, and sporulate efficiently. Okay, otherwise you couldn't afford to deliver the appropriate dose, okay? Not 100 times less than what works in the lab, but to be able to afford to use what actually works. And it must have in vitro activity against common enteric pathogens, and they must be GRAS, generally recognized as safe by, in our country, the Food and Drug Administration, AFCO, and so forth, and similar regulatory agencies around the world. The criteria for the final isolate selection that we used was that isolates must germinate within the avian gut. The specific isolates were selected to complement one another. We ran a number of studies, and we found the same thing that we did with the lactic acid bacteria. Some were complementary, and some were antagonistic. The final formulation was selected for in vitro and vivo activity against selected enteric pathogens. And I'm going to skip over some of the laboratory studies. I'm going to show you a little bit, but then I'm going to show you field studies that have been done more recently. Just to give you an idea of how we screened for these guys, in vitro antimicrobial activity was done using an overlay system. A number of pathogens are used. Basically, you plate the bacillus colonies, overlay with a soft agar overlay, and you're able to see for those candidates that are actually competing with the pathogens, you can see these zones of inhibition, okay? And very clearly, some of these compete very nicely. We also looked for the ability to compete with pathogens in an organic matrix environment. And that's really important because if you want to kill pathogens in a very in the absence of large amounts of organic matter, it's fairly easy. Even a little bit of chlorine will do that. But once you start adding a organic matrix like poultry feed to the equation, many of these compounds that have antimicrobial bacteria also bind to proteins and lipids and so forth within the diet, and they're not active anymore. So we look for things that actually would work in the presence of feed in vitro. So this is another reason to throw things out. We were not trying to keep one, but rather to eliminate things before we went to animal testing. We needed to have generally recognized as safe status. There's only a few groups within the bacillus that fits that characteristic, so we threw them out if they were not generally recognized as safe. We only chose non-hemolytic strains because the hemolysins in many cases do damage. Some of the candidates that we produced actually reduced performance in the birds in our studies, and we found that some of those were because they were producing hemolysins. We also tested them for the production of enterotoxins using a Bacillus cereus kit, because some generally recognized as safe bacillus can actually produce enterotoxins if they have the right plasmids. Identification is really easy with the bacilli. Hemolysis is really easy to see. And we focused heavily on spore yield because we threw away candidates if we could not get high spore yield because they could never be commercially useful, because you'd never be able to afford to produce them at levels that were needed. There's extensive literature with the few isolates that have been shown to actually have benefits in poultry for bacillus, that you need about 10 to the 6. That's 1 million colony forming units per gram of finished feed. We're talking 10 to the 12th bacteria per ton of feed, okay? That's a lot of spores, okay? And the literature suggests that that's true. Our data supports that that's true. You can do the experiments and you can show that they work at 10 to the 6th or 10 to the 7th, and then you can sell the product really cheaply if you do 100 times less, okay? And that's a trick that doesn't make any sense to me. We're talking very high numbers of spores to be able to get these, have the right spores and delivered at a very high number. We actually developed some proprietary methods to amplify very high yields, and that was licensed to Pacific Vet Group. And we also screened by pasteurizing samples initially for 70 degrees C for 15 minutes, but the candidates, before they went into birds, had to survive 100 degrees Celsius, boiling, for 10 minutes before we would even take them to the animal trials. So we were throwing isolates away in this whole process. Okay. One of the models that we use is a horizontal transmission of a very aggressive Salmonella enteritidis strain in our laboratory. And here you can see the effect on the liver spleen colonization with the contact birds that were treated versus the untreated, and the colonization of the ceca in the treated versus the untreated. That's very encouraging when this happens in a very short period of time. And the numbers that are actually in the treated birds are markedly less. That's a 2-log reduction. That's a 100-fold reduction in numbers of Salmonella, even in the infected birds. We took this to our necrotic enteritis model. OX-CTA are regarded as an absolute prerequisite for this, and then high levels of Clostridium perfringens challenged. And basically, we're looking at a challenge between 22 and 26 days, and by 29 days, we're measuring body weight gain, mortality, and lesions. And we compared again against one of the most popular antibody growth promoters, which I actually think is a good product in these studies. This is crazy to think that you can match or even beat an antibiotic growth promoter, but we did it anyway. And we're looking here at body weight gain. The controls, very significantly different with bacitracin, 22% of the controls died in this study. Untreated controls died. Bacitracin, with this, the one I showed you before was a very high challenge, but from 22%, bacitracin protected from necrotic enteritis. That's what we see in the field most of the time. And spoilin, 0% mortality and identical body weight statistically. Looking at the lesion scores, as you would expect, bacitracin produced a tremendous drop in necrotic enteritis lesion scores, as did spoilin. This is a field trial where we actually, this is the same type of system that that I referred to earlier, where we can place animals right down the center of the house in individual pens in the real world environment and look at performance. And we compared in this study the body weight gain based on histostat, an organic arsenical that it's considered one of the top-notch products